All right, so you used the slides to fill in the information, and again, the intent is now you can pay attention and be more actively engaged than worrying about writing down words, okay? So that's my expectation, not that you're playing football on your phone, not that you are scrolling whatever social media you guys scroll through anymore, okay, that we're paying attention to this. And I have a fair amount of examples, and I'll try to keep this light and entertaining as much as I can. But we talk about parts of the cell. Now, what are some basic things that you need in order to live, survive? Let's talk relatively short term. Food, water. What's that?
Sleep.
Yeah, sleep. Okay, if you go a long enough period of time, yes, you do need some sleep, but what else? Food, water, oxygen. Okay. Other things, yeah, shelter, clothing. Those are all some things that make life a lot easier. But if we talk about those basic three things of air, oxygen, oxygen, water, and food, the reason you need those things to live is because your cells need those things to live. So your cells need oxygen, your cells need food, your cells need water in order to survive. So we got to get all that stuff to your cells, and then once they get to your cells, it needs to go into your cells. We also got to get rid of waste through our cells. So the mechanism that most of our stuff gets to our cells and leaves our cells is with the circulatory system. A lot of our stuff is pumped with blood, and once it reaches that cells, then this is what we're talking about. So how does it enter the cells and how does it leave the cells? So when we talk about things entering and leaving the cells, the cell membrane is the thing that kind of keeps some things in and some things out, and it really regulates that. We've often just drawn this as a single line around an object, but there's really more to it. So we talk about these, the molecule, if we were to zoom in on a cell membrane or a plasma membrane, we get these things, and this is called a phospholipid. Now, phospholipid is made up of two things. It's made up of a head, the red part, that loves water, it's hydrophilic, and then we have a tail that's a lipid, and it hates water. So this is like oil, this likes oil, this likes water. Oil and water don't mix. But when you put these kind of together, they form this, what we call a bilayer. It's a that's a phospholipid, that's a phospholipid, that's a phospholipid. And we form two layers of them, so they call it a phospholipid bilayer. And these things are just kind of connected together and encapsulating each other. And there's also some proteins, and on the board we have, Mr. DeBoer drew a different picture of our phospholipid bilayer. And there's some protein channels, and we have some carbohydrate chains, and we'll talk about those a little bit later. But we kind of collectively, we call this a mosaic, okay? And a lot of times we refer to it as a fluid mosaic model because this is very flexible, and it moves and it flows, and it conforms to the shapes. So the cell membrane is a structure that all cells have. All cells have a cell membrane. All cells have genetic information. All cells have mitochondria. All cells have ribosomes. Not all cells have a nucleus and some other parts, right? But they all have a cell membrane. So when we talk about, again, phospholipid bilayer, this is like the picture Mr. DeBoer drew on the board. We have our phospholipid bilayer with these things called channel proteins or protein channels or transport proteins. They're also called that allows some things to move through, and we have these things called carbohydrate chains. Now the carbohydrate chains are responsible for some cell-to-cell signaling, and it's like when two cells are in contact with one another, they can communicate through some of these carbohydrate chains. This is also how we sometimes talk about food carbohydrates entering, but this cell-to-cell signaling is important. So the signaling is kind of like, should we let this thing or not let this thing in? So with SARS-CoV-2, the virus that causes COVID, it has those very traditional spike on them, right? We've seen those pictures of those spikes, and they call them spike proteins. And what happens with those spike proteins is when they bind to the cell membrane, those spikes signal to the cell, and the cell's like, yeah, you're good, come on in, and encapsulates it, and the virus enters in your cell. and then it hijacks your ribosomes, and it actually uses the ribosomes to mass produce new viruses, and then they export them and then continue to spread the virus throughout your body. But it's because those spike proteins signal to the cell membrane and say, yeah, come on in, you're good. So if you've gotten COVID, you have some antibodies now floating around in your body. If you got vaccinated, you have antibodies floating around in your body. And the idea is those antibodies will actually bind to the spike proteins on the virus, which changes the shape of the virus. So when the virus comes to the cell membrane, it doesn't match. And it'll be like, nope, you can't come in. I don't recognize you. You don't recognize the spike proteins. So if they don't enter the cell, then that virus can't replicate in your cells. And you, in theory, I gotta stop saying in theory. The idea is then you shouldn't get infected with virus or then get COVID. That's what they talk about when you get natural immunity from having COVID, or if you got vaccinated, it changes the shape of the spike proteins, and that cell-to-cell signaling prevents it from entering the cell, actually. So all cells have a cell membrane. Some cells have a cell wall, like plants, algae, fungus, and many other prokaryotes, bacteria cells. Not all cells have cell walls, but some do. It's another layer that some of these things have to get through. Some things go through better than others. And in plants, it tends to be made out of carbohydrates called cellulose. Cellulose is what I'm knocking. Cellulose is often referred to like as fiber. Okay? If you have a lot of fiber in your diet, you're eating a fair amount of cellulose. And what that is, it's the cell walls is really what you're eating. Our body does not do a good job of breaking down cellulose. Our body does not digest fiber very well. It has an important role. We need fiber in our diet, but we don't get nutritional value from cellulose. Corn, for example, a kernel of corn has a fair amount of cellulose in it. We don't digest corn kernels very well. Okay? They taste good, but we don't get much nutrition out of them, us. Okay? So when we talk about cellulose, it's just really remnants of the cell wall. So like wood is a good material that we can build out of, but what it is cell membranes, the plasma membranes have like shriveled up. All the stuff inside the cell has like dried up and died. All we're left with is the cell walls. You know, a lot of cell walls for that matter. So things have to enter and leave. So when we talk about things entering and leaving the cell, we talk about this thing called diffusion. So diffusion is the movement of substances from an area of high concentration to an area of low. High to low, high to low, high to low, high to low. Diffusion is a random movement of molecules from high to low. Because molecules, and my corks are molecules, are in constant random motion. It's called the kinetic molecular theory. And the idea is molecules at the atomic level are doing this. Sometimes they're doing this, like a solid, like your desktop might be doing this, where the air around us is doing something more like that. They're moving much faster, they're further apart. But when we talk about diffusion, diffusion is the area of movement of molecules from high concentration to low concentration. So if I take this Febreze Air Forest and I spray it up there, I smell it. It smells like forest. You guys smell it? How do you smell it?
No.
I smell it. Just wait. Okay? So if I spray, I sprayed a lot, right? Right here. What's true about the concentration of Glade Air Forest particles right here compared to where you are? High or low? Right here. High, lots of it, right? What about where you guys are? Low. What's going to happen to these particles given enough time? Anybody smell it yet? Front row, you smelling it yet? Okay. We're getting back. How far back do we smell it? Raise your hand if you smell it. Okay. I'll ask you again in a second. So what's happening is they're slowly going from high concentration because these particles are doing this. But there's a high concentration here and there's low, and it's going to spread out throughout the room. And given enough time, they will reach something called equilibrium, where the concentration of those air particles, or these air freshener particles, are going to be equal throughout the room. Now what could happen is they become so dilute, or the concentration becomes so low, that we then don't smell it. Or with air freshener, it might settle, and then because they aren't floating around in the air, that's why we're not smelling it. Now, and raise your hand. Now do you smell it? More people smelling it? Further, I see a little bit further back starting to smell it. Okay. So the idea is we go from high concentration to low concentration. Now, why are those air particles, or those air freshener particles moving? Because they're constantly bouncing around. I like to think of movement of molecules in diffusion like a mosh pit. Okay? Maybe you've been to a concert that has had a mosh pit. Maybe you've been part of that mosh pit. Maybe you haven't been part of that mosh pit. Maybe you know what a mosh pit is because you saw it on TV. Regardless, a mosh pit is this thing, typically at a concert, that people are generally down in front of the stage, having a good time, but just going crazy, really, right? That's probably a good way to describe a mosh pit. And what happens is you are just kind of shoving each other all over the place, and you're getting bounced from one spot to the next, to the next, to the next, to the next. Okay? Big potential health risk associated with a mosh pit, like you could fall over and get trampled, or get hurt or stepped on, whatever. Let's talk about this, okay? So there's a giant mosh pit right here of air particles. And what happens is, because they aren't in control of their body, they're just getting shoved around. What eventually happens is it spreads out and it goes to you. It doesn't take up any energy. No energy is required in diffusion from high concentration and low concentration, because they're in a constant random state of motion. Same thing with a mosh pit, right? So, if we have an area of high concentration and these molecules are allowed to go across the cell membrane, they tend to go from areas of high concentration, there's no, so this is low, they're going to go across the cell membrane until a point where they reach equilibrium, where the concentration outside and inside are the same. And we talk about there's no net movement of molecules. There are still some going in, but on average, for every one that goes in, one would go out. They average out. So eventually, this air freshener, I still smell the air freshener up here, is going to reach a state of equilibrium throughout the room, and it's going to be the same concentration there as it is there, as it is there, where it was really high compared to there. So diffusion, I like to think about as a mosh pit. Sometimes I'm not in my classroom when you guys are ready to learn biology. So this is our classroom door. You guys are outside, bio mosh pit, right? I've seen them. I've seen it. I've been in, like, walking through the back, and I'm like, oh my goodness, I gotta open up the door because there's a biology mosh pit going on out here, and they'd be like, let me in. But the door's closed, right? And you can't get in because the door is closed. And then I want you guys to learn biology, so I very quickly open up the door. Now what happens because mosh pit's going on out there? A lot of control over your body? Do you control where you're moving in a mosh pit? A little bit. For our analogy, no, right? The person next to you shoving you, just bouncing off each other. Random, constant motion. So when I open that door, what happens to some of you? Will some of you randomly bounce into the room? You will, right? And there's only one of me in here. There's 25 of you out there. I'm doing my own mosh pit in here, just for the record. Like, I can't wait to teach biology. I'm doing my own mosh pit. Okay, mosh pit, mosh pit. But when I open the door, your concentration is much higher. So you guys tend to diffuse into the room to kind of match out. Now, if we were like equal confined spaces, right? So like we box off like 20 feet by 20 feet and 20 feet by 20 feet or 10 by 10. That'd make this better, an analogy a little bit better. But the idea is, like, when I do, some of you are going to fall in. Now what happens to some of you is you're out there and you're like, oh, biology, I can't wait to get biology. And you're just getting bounced around. And you're going to come like, oh, I'm going to biology. And they're like, oh, I miss biology. I can't get to biology. It's mosh mosh pits. You can't control it, right? But that's the idea of movement of molecules. Constant random motion. You were so close to making it to biology, and you ran into the door. Okay? Now, unfortunately, for some of you, you've made it in. No, that's the good part. Fortunately, some of you have made it in, but you're still mosh pitting in here. And eventually, like, I think you said 25 and 1, so 26 of us total, right? Our equilibrium point would be 13 to 13, right? But you're still mosh pitting in here. Some of you have made it. Yes! But you're still in a mosh pit. So you don't have control of your body, and so you're going to be like, no, I want to be in here. Ah! And you can't help it because it's a mosh pit. But in equilibrium, some of us are going to go out into the mosh pit, and some people are going to be coming in. But we say there's no net movement because on average, for every one person that enters the room, on average, one person would exit that room. Now, if I took a picture, once we've reached equilibrium, at some point it might be 14 and 12, but then given like a minute later, it might be 15 and 11, right? So they are going to move, but on average, we say that there's no net movement. It balances out. They're still moving. They're still doing this. Hey, Barb.
Hello, excuse me.
Yeah.
Rivers Astro?
Yep.
Caden?
Nope.
Harold?
Yep.
Nathan?
Yep.
Paige?
Yep.
Thank you.
Mm-hmm. Yes. Okay, so mosh pit, we're doing our mosh pit, right? And eventually we reach what we call equilibrium, where some of us exiting and some of us leaving are going to end up being the same. When we talk about diffusion, there's this thing called concentration gradient. Diffusion requires no energy, and the bigger the difference in concentration, the more movement we're going to get. Okay, the more movement we're going to get. The analogy we like to use is a slide. The steeper the slide is, the faster you go down the slide, right? Okay, this is like a toddler slide. That for a toddler is really fun and exciting because they go down like 1 foot and you're like, whoa, good job. So that's what you do for toddlers because that was a big deal for them to go down that 1 foot slide. But for us, much more advanced sliders, we like a little bit more thrill. We like, the 6 foot slides. Maybe you go 10, maybe it's crazy, right? But the idea is the steeper that slide, the faster you end up going. Same thing when we talk about concentration gradient. The bigger the difference in concentration, the quicker that movement is going to happen. So 25 people outside mosh pitting versus me in here. Open that door. Early on, we're going to get a lot more of you coming this way into the room than going out. But as we get closer to that equilibrium point, the amount of people bouncing in is going to slow down. It's more like the toddler slide. Not as fast. There's still movement. There's still a net movement, but it's not as quickly. Okay, so we talk about slides being the concentration gradient. The taller the slide, the faster you go. The bigger the difference in concentration, or the steeper or the larger the concentration gradient, the faster that movement goes. So some things are too large to fit through the cell membrane, or they're too strongly charged. So then we got to use like these things called like transport proteins or protein channels to help out. So we use these terms called permeability. So something could be permeable, it could be impermeable, or semi-permeable, or sometimes instead of saying semi, we use selectively permeable. So if I have that door and I have the door closed, is that door permeable or impermeable? And maybe before we do that, let's talk about the term permeable. If that door is open, the door is permeable to me, right? I can permeate through. Not perfect, but for our analogy it works. I can permeate through, I can go through it. If the door closes, is that permeable or impermeable to me? I tried it last time. It didn't work. I can try it again if we really want.
Yeah, try it again.
Okay, so if I take the door right now, permeable. I should make sure I'm on this side because it locks if I'm out there. Okay, I can walk through. Okay, but if, let's see if the door is permeable. Nope, still not permeable to me. Impermeable, right? Is it impermeable to all things though? Can anything get through this door? What? Where? Yeah, maybe a bug could crawl underneath. What else? Air, right? It's not perfectly sealed. Light, right? So some things can come through. It's permeable to light, air. Someone said water. Yeah, water running through the door. Okay? But it's impermeable to me. So then we refer to that as semi-permeable or selectively permeable because it allows some things through, but it doesn't allow other things to go through. A cell membrane is semi-permeable or selectively permeable. Screen door, probably a better example of semi-permeable, right? So I have a screen door. I close my screen door. What is my screen door let through? What's that? Air. That's probably why we have a screen door, is to let some air movement go through. Light. What happens if it rains? Unfortunately, it will let water in. But what's the intent of a screen door? To try to keep bugs, birds, squirrels out, right? So it's semi-permeable because it lets some things through, but not other things do. And same thing with a cell membrane. So now we take this idea of mosh pit diffusion, concentration gradient, and we can apply it to a very specific thing. Water is very important for life. Life as we know it needs water. Life as we know it needs water. So it's so important. We talk about diffusion of water and we give it its own term, and it's simply called osmosis. Osmosis is the diffusion of water. All the same characteristics apply. It's still water. It's just a water mosh pit now, right? So where is the concentration of water highest? Where is it lowest? And it flows from high concentration to low. It goes down the slide. It goes down the concentration gradient. The bigger the difference, the faster it is, the steeper that slide is. And it's going to keep going until it reaches equilibrium, and no energy is required. It's a mosh pit, bouncing all over the place. Once they reach the same concentrations, then it reaches that equilibrium, which we say there's no net movement of water molecules. There's no net movement of water molecules. So if this is a beaker that has a semi-permeable membrane that lets water molecules through but not the other stuff, the concentration is higher on this side, right? There's more dots than there is on this side, so it flows from high to low concentration. Okay, I spoke too soon. The dots in this picture don't represent water molecules, they actually represent like solute molecules. So we could say like, well, is there more, the space between these dots, sorry, is water, the space between these dots is water. So where is there more water? There's more water over here and comes across. Which leads us to these terms. And these terms are the things that people struggle with. But when we talk about osmosis, we talk about the movement of water and the effect that it has on cells. So we talk about these things called solutions, and a solution is a mixture. It's a liquid mixture. And most of the time when we talk about a liquid mixture, we're talking about a mixture of something in water. Okay? So there's two parts of a solution. We have something called the solute and the solvent. If we're talking about water, water is the solvent. It's the thing that dissolves stuff, okay? So our solutions are often pretty basic: sugar water, or salt water, or Kool-Aid, or whatever. But something with water. So water is our solvent. Water is also known as a universal solvent. In reference to it, dissolves a lot of things. So then we talk about, well, what happens to the water? So we talk about what we care about in cells is which way the water flows. Okay, is water flowing into the cell? Is it flowing out of the cell? Or is it flowing equally in and out? Is it at equilibrium? Unfortunately, the terms that we use, called hypertonic, isotonic, and hypertonic, don't refer to the water, which is what we care about. It refers to the solute. It refers to the salt particles. It refers to the sugar particles. So that's what we have to kind of look at. So when we talk about, like, if I had two containers, like I have one beaker, I have a beaker of water, and in that water is water. And then I have another beaker that I put a bunch of salt in, and then I fill it to the same height of water. Okay, so this is nothing but water. This is salt and water. In which beaker, A, all water, or B, salt water, is there more water molecules? Only water, or the mixture of salt and water? Only water, right? Does salt take up space in a container? Yeah, like if you just take your salt shaker and go sprinkle, sprinkle, you're not gonna notice it. But if you fill like a lot of salt, it takes up space, okay? So in beaker A was only water, there's more water molecules. Water molecules are higher here than they are here in the salt water, okay? So the idea is if I have salt and water in here, I have a lower concentration of water, but if I talk about the salt, I have a high concentration of salt. Okay, this one has a very low concentration of salt, or in my example, no. Okay, I take a pinch of salt, there, I put a pinch. This is a little bit of salt, this is a lot of salt. My salt concentration is very low. And then we use these three prefixes, hypo, hyper, and iso, to reference the solute particles, so the salt. So use the words like you know, hyper, hypo, and iso. So hypothermia, what's hypothermia? Your body temperature is cold or below what it should be. You don't fix it, you'll die. Okay? Hypo means below. Your body temperature is cold, you're below temperature. Okay? We don't use the term. We could say you're hyperthermic. We normally would call it like heat exhaustion or heat stroke, something along those lines. But above, a different way to kind of think about this is, if you were to very politely say that, boy, your child is very active, has a lot of energy, because we don't like to use it, but what we're really trying to say is your kid's kind of hyper, and woo, he needs to calm down a little bit, right? Hyper means above normal, kind of, right? That's what hyper means. The child's a little hyper, it means above normal energy levels. And in math, we talk about different types of triangles. An isosceles triangle has two sides that are the same. Right? Isosceles triangle is they have two sides that are the same or equal. So if we use those kind of prefixes to kind of help us, is it hyper, hypo, or iso? Is it above, below, or the same? We can then kind of help identify what's going on. What we're talking about is the salt, or the sugar, or the iodine, or the starch, or whatever. We're talking about the solutes, we're talking about the salt sugars, not the water, but what we care about is the water. So if my salt concentration is very high, then it's going to be hypertonic. If my salt concentration is low, it's hypotonic. If it's the same, then it'd be isotonic. And we always reference it compared to the cell. So is the blood or the solution surrounding my cell above, below, or the same compared to the cell? Because that's what we care about. We care about cells. Then we have to kind of reverse, say, okay, well, I have above average salt solution, which then means my water concentration is low, which then means my water is going to flow in, it's going to flow out. So these terms get slightly messy and confusing. And that's what these are talking about. So isotonic, in the picture, the sugar molecules are represented by these green dots. I have the same amount of green dots here as I do here. They're the same inside and out, therefore it's isotonic. I have 1 versus 1, 2, 3, 4, 5, 6. Outside is lower than inside, is hypotonic. Over here, I don't know, I have lots versus little. I have higher than outside than inside is hypertonic. But what's going to happen is the water is in reverse. So if this is low outside of salt, that means this is high water, which means my water is going to flow in. This is lots of salt, a little bit of water, so my water's gonna go out. What happens if you're dehydrated? Your cells lose water, right? It's flowing outwards. And with plants, we have something called osmotic pressure. And what happens is that, remember that, what's that? Oh gosh, the thing inside cells, it's large, it's central, it's a storage thing. Vacuole. Yeah, it's a vacuole, right? So what happens if your plants don't have enough water is that vacuole starts to shrink. And as that vacuole starts to shrink, your cell membrane actually pulls away from the cell wall, but your cell wall is still there. And then it lacks something called osmotic pressure, and your plant starts to wilt. But then what happens when you water your plant, now you have a high concentration of water, the water is going to flow into your cells, that vacuole is going to swell, and it's going to push against the cell wall, and your plant will not be wilted. Plants have the benefit of having that rigid cell wall to kind of confine that cell membrane. It kind of hits that wall and it stops. We don't have a cell wall, so what could potentially happen if we got too much water in our cells? The cells could get bigger and bigger, but eventually what happens to a water balloon if you put too much water in it?
Pops.
It could pop, and your cells could potentially pop if you drank too much water. It is a thing. People have died of too much water intake. It's not super common. You go to the bathroom, it should help get rid of excess water. Okay, but that's the idea. Okay? Have a great day.